Hello everyone, I am K. Devani, Assistant Professor of Microbiology from Government Degree College for Women, Nalgonda. In this video, I am going to explain the topic on cells of immune system. The successful immune response to a pathogen always depends upon the interactions between the various cell types that may be of innate immune cells which form a first line of defense against a pathogen, and the antigen presenting cells that communicate the infection to the lymphoid cells. They coordinate the lymphoid cells which can coordinate the specific immunity, that is adaptive immunity, and generate the memory cells which can prevent the future infections. So, the coordination of various diverse cell types is required for a full immune response. Now, I am going to explain the cells of immune system which have a role in the innate immunity as well as in the adaptive immunity. In this video, I am going to explain up to the cells of myeloid lineage. The cells of lymphoid lineage will be explained in the next video. The first one is hematopoiesis, and the second one is cells of myeloid lineage. The third one is cells of lymphoid lineage and the dendritics. First one is hematopoiesis. Hematopoiesis is a process by which hematopoietic stem cells, H means hematopoietic, S means stem, C means cell, hematopoietic stem cells differentiate into mature blood cells. The process by which the hematopoietic stem cells differentiate into blood cells is called as a hematopoiesis. Stem cells, they have two abilities, that is self renewal and another property is differentiation. Self renewal is the ability to regenerate and differentiation is the ability to be differentiated into various cell types. Embryonic stem cells, they are pluripotent. That means embryonic stem cells, they are pluripotent means they are able to generate any specialized cell. They are able to generate every specialized cell type in an organism is called embryonic stem cells. There are stem cells, another category is called adult stem cells, which are called as ASC. They have the capacity to give rise to cell types which are specific to a particular tissue or the cell. That means adult stem cells are already committed to form a specific cell type. Now, the hematopoietic stem cells, they are considered to be the adult stem cells and they can differentiate into all types of blood cells. Hematopoietic stem cells are a type of are considered to be adult stem cells because they can differentiate into all types of blood cells. The process by which HSCs differentiate into mature blood cells is called as a hematopoiesis. Hematopoietic stem cells are rare and fewer in number. Their numbers are controlled by the balance of cell division and death. Under normal conditions where the immune system is not being challenged by a pathogen, hematopoietic stem cells are in quiescent stage, that means in dormant state. Small number they divide and they produce daughter cells. Small number of hematopoietic stem cells they divide, and some cells they have the self renewal ability, that means they can regenerate, but some cells they lose their self renewal capacity and they are become more committed to a particular blood lineage and they are called as progenitor cells. Hematopoietic stem cells, some they lose the self renewal capacity, they are called as progenitor cells, and some cells they retain their self renewal capacity. As the organism ages, the number of hematopoietic stem cells decrease. It is an undifferentiated hematopoietic stem cells. They would not express any surface markers. The surface markers are specific to the mature cells. The hematopoietic stem cell that is differentiated into a cell that loses the self-renewal ability and makes this hematopoietic stem cell, that is called hemocytoblast. It produces one of two lineage commitments, that is, one is called as a common myeloid progenitor, another one is called as a common lymphoid progenitor. Common myeloid progenitor and this one is a common lymphoid progenitor. This common myeloid progenitor, it gives rise to the cells, myeloid cells and erythroid cells. The myeloid cells here comprises of granulocytes, megakaryocytes, mast cells and myoblast. This myoblast, it gives rise to the granulocytes and agranulocytes of W.B.C. They are basophils, neutrophils, eosinophils and monocytes. Erythroid cells, that they gave rise to erythrocytes, and megakaryocytes, they give rise to fibroblasts or thrombocytes. Now here coming to the lymphoid progenitor, common lymphoid progenitor, that is CLP. It produces lymphocytes and natural killer cells. They are also called as LGL. Natural killer cells means large granular lymphocytes, and the small lymphocytes, they are categorized into T lymphocytes and B lymphocytes based on their surface markers, and these B lymphocytes, they will be transformed into plasma cell after come in contact with the antigen and release the antibodies specific to that of particular epitope. So this is about the hematopoiesis, origin of different kinds of blood cells. And the dendritic cells or cells, they are produced by both lymphoid lineage and myeloid lineage. Both lymphoid and myeloid lineage, they give rise to a dendritic cells which have the diverse features and functions that play an important role in initiating the adaptive immune response. The blood cells starts with the multipotent hematopoietic stem cells, hemocytoblast, which produce two kinds of progenitor cells which lost the self-renewal capability. They are common myeloid progenitor and common lymphoid progenitor. The common myeloid progenitor, it produces cells is the megakaryocytes and erythroid cells and myeloid lineage, and the myeloid lineage, it produces again granulocytes and agranulocytes, and common lymphoid progenitor, here it produces natural killer cells which are also called as large granular lymphocytes and the small lymphocytes. The lymphocytes are two types here, the T lymphocytes and B lymphocytes, and the B lymphocytes are transformed into plasma cells on contact with the antigen. Now moving on to the cells of myeloid lineage. The cells of myeloid lineage include the granulocytes, which are the neutrophils, eosinophils and basophils. Mast cells are also, they share the features with the basophils. Granulocytes, they are the front lines of attack. They form the first lines of defense during the immune response. Granulocytes are W.B.C. They are W.B.C. They are classified into neutrophils, eosinophils and basophils and mast cells. They are classified based on their morphology and staining. Cells possess the granules based on the staining properties of those granules and the cell morphology. These granulocytes are classified into neutrophils, eosinophils, basophils and mast cells. First one, we will see the neutrophils. Neutrophils, here they constitute about 50 to 70%. They are more in number than the eosinophils and basophils and mast cells. Neutrophils are multi-lobed, and these lobes are joined by a strands of chromatin, that is genetic material, and it contains a granules. These granules are called as primary granules and secondary granules. Granules rich with enzymes. Neutrophils, they show the process of phagocytosis. After differentiation in the bone marrow, these neutrophils, they are released into the peripheral blood and circulate for 7 to 10 hours before migrating into the tissues. So, these neutrophils, they circulate in the blood for 7 to 10 hours. They have a lifespan of few days in the tissues. Later they reach the tissues, they have a lifespan of only a few days. In response to the infection, the number of circulating neutrophils increases, that is called as a leukocytosis. Leukocytosis is the process of increasing the number of neutrophils, particularly that occurs during the infections. So, that is called as a leukocytosis, and they are the predominant cell types in the acute inflammation, and they are recruited by means of attracting to the chemokines to the site of infection. They are thought to have any role in the specific immune response also. Again, about the neutrophils, they are called as a polymorphonuclear leukocytes, and they are more in number in the circulating leukocytes compared to the eosinophils and basophils, and they contain the granules which possess the enzymic activity, and they have the process, they show the process of phagocytosis. The number of neutrophils increase during infections, that is called as leukocytosis, and they are the primary predominant cell type in the acute inflammation, and they are recruited to the site of infection by attracting to the chemokines. The process of movement of these cells towards the infection is called as a chemotaxis, and these have a multi-lobed, the lobes are joined by a strands of genetic material. These neutrophils generally after differentiation, they are released into the blood, and in the blood they circulate for 7 to 10 hours, and later they reach to the tissues, they migrate to the tissues. In the tissues they have a lifespan of very few days. Next one is eosinophils. Eosinophils are called as motile phagocytes. They have motile, they can migrate from blood into tissue spaces. They are bilobed, and stained with eosin, and they are called acidophils. Because the cytoplasmic granules, they have the affinity to acids, that is why they can also called as acidophilic, and the cytoplasm is filled with many granules, approximately 200 granules, which contains the enzymes and proteins. But though they show the phagocytosis, it is of lesser importance, but they are found majorly in the allergic inflammation and parasitic infections. Primarily they are present in the tissues rather than blood stream. The granules here are again two types, smaller granules and large granules. The smaller granules are in round shape, whereas larger granules are in ovoid shape. These granules, they contain a variety of hydrolytic enzymes. These enzymes, they bring about the extracellular killing of large parasites. Eosinophils, they also secrete cytokines. All these granulocytes, neutrophils, eosinophils, basophils, they secrete cytokines. The cytokines that regulate both B cells and T cells, and which influence the adaptive immune response. Eosinophils also contribute to asthma and allergic symptoms. The majorly found in the inflammation, allergic inflammation and the parasitic infections. The third one is the basophils. They are found in the blood. It is about only 0.5 to 1%. Eosinophils generally they constitute about 1 to 3%. The basophils number is very less, that is less than 1%. And basophils are discovered by Paul Ehrlich in 1879. He named them as mast cells, which are present in the tissues, for which he received the Nobel Prize. And basophils also contain granules. The granules contain heparin, and it is also nucleus also has two lobes, and the granules contain heparin serotonin, histamine and other enzymes. They stain blue with basic dyes. They are non-phagocytic. In these granulocytes, basophils are the non-phagocytic granulocytes. Basophils release the contents of the granules in response to binding of circulating antibodies, particularly that is IgE. When the circulating IgE antibodies they bind, it leads to the degranulation of the basophils that release the proteins that are present in them, which are one of the important best known protein here. It is a histamine, which is the best known protein in basophilic granules. That histamine increases the blood vessel permeability and smooth muscle contraction. This blood vessel permeability and smooth muscle contraction, it promotes the blood flow to the tissues. Other cells that are present in allergic symptoms and cause allergy, hypersensitivity. And the another protein that is found in these granules is heparin. It prevents the blood from clotting. Basophils also secrete cytokines. So, basophils they constitute about less than 1%. They are non-phagocytic and they stain blue by basic dyes, and they were discovered by Paul Ehrlich, and he named mast cells that are present in the tissues. Basophils they release the contents of the granules in response to the binding of antibodies IgE. Histamine is one of the best known protein that increases the blood vessel permeability and smooth muscle contraction that promotes the blood flow to the tissues. And another protein that is present in these granules is heparin that prevents the blood from clotting. The another one is the mast cells. Mast cells are released from the bone marrow into blood as the undifferentiated cells. They mature only after they leave the blood. They are found in variety of tissues including skin, connective tissues of various organs and mucosal epithelial tissues of respiratory tract, digestive tract and genitourinary tract. Like circulating basophils, these cells, mast cells also have the large number of cytoplasmic granules. These granules they contain histamine and pharmacologically active substances. Basophils and mast cells, they share the features and they play an important role in development of allergies. Basophils and mast cells they share the many features and they play an important role in development of allergies. Another category of cells that are produced from the myeloid origin, they are antigen processing cells, which are called as APC, or antigen presenting cells. They arise from the myeloid progenitors, which include these antigen processing cells majorly include monocytes, macrophages and dendritic cells. These myeloid APC, they generally serve as a bridge between the innate immunity and adaptive immunity. They serve as a connecting link between the innate and adaptive immune systems. Antigen presenting cells, they come in contact with the pathogen at the site of infection and they are processed by these cells, and these cells they present the antigen to the T cells. First one is monocytes. Monocytes are 5 to 10% of WBC. These are agranulocytes. So, agranulated, single lobe nuclei, nuclei single lobe, that is unilobe nuclei. And monocytes, these cells they migrate into the tissues and they contain the granules, the eosinophilic granules are present, and these migrate into the tissues and differentiated into phagocytic cells. They are macrophages including dendritic cells. So, monocytes when they migrate into the tissues, they differentiate into macrophages and dendritic cells. During hematopoiesis in the bone marrow, granulocyte, monocyte progenitor cells, they differentiate into monocytes. These monocytes, they leave the bone marrow and they enter into blood, where they differentiated into mature monocytes. The monocytes are two types, inflammatory and patrolling. Inflammatory monocytes, they enter into the tissues very quick in response to the infections. Patrolling monocytes, they are in small group, they slowly move along the blood vessels and they serve as a reservoir for the tissue monocytes in the absence of infection. Monocytes in circulation have a half-life of 3 days. Monocytes, they migrate into tissues in response to infection and differentiate into macrophages. These macrophages, they survive for many months. Differentiation of monocytes into a tissue macrophage involves number of changes. Those changes are the cell increases in size by 5 to 10 fold and the intracellular granules that are present in these monocytes, they also increase in number and it acquires the increasing phagocytic ability. There is a change in the increase in the phagocytic ability and produce high levels of enzymes, hydrolytic enzymes, and these macrophages, they are now able to secrete a variety of soluble factors. These are the changes that would be observed when a monocyte is differentiated into a macrophage. The changes would be the increase in the cell size, the granules and the enzymes, and the phagocytic ability will be increased and they are able to secrete the variety of soluble factors. Now the macrophages serve different functions in different tissues and functions in different tissues named according to their location. Examples like in the lungs, they are called as alveolar macrophages. In connective tissue, they are called as histiocytes, and liver, they are called as Kupffer cells. Brain, microglial cells. In the kidney, mesangial cells, and in the bone, osteoclasts. So they are named differently based on the presence of different tissues. And macrophages, these macrophages, they are present in the tissues for a long period and they play a role in regulating the repair and regeneration. Some macrophages, they are present in the tissues for a long term and they play a role in regulating the repair and regeneration. Other macrophages, they participate in the innate immune response and the inflammation. Macrophages in the innate immune response and inflammation, they contribute to the clearance of pathogen and also they serve as an antigen presenting cells that can activate the T cells. So the macrophages here have different roles. They are the first line of respondents in the infections and they serve as an antigen presenting cells. The antigen presenting cells, they can activate the T cells and finally they contribute to the clearance of the pathogen. The mechanism of phagocytosis is explained in the phagocytosis. Here the clinical significance of macrophages. The phagocytosis is an effective defense against many of the microorganisms, but still some microorganisms are resistant to the degradation by the phagocytosis. Such examples include tuberculosis, brucellosis. These organisms are resistant to digestion and they multiply within these cells and they are transported to different locations in the body. Macrophages express surface receptors. The surface receptors are Fc part of immunoglobulin and the complement proteins. Complement receptors for complement proteins such as C3a and C5a. So, when the immunoglobulin and complement proteins they bind to these macrophages, the phagocytosis process is enhanced, that is called as opsonization. The immunoglobulin and the complement proteins serve here as a opsonins. The features of activated macrophages is activated macrophages are not antigen specific and the functional efficiency of macrophages can be increased by the factors like lymphokines and complement and interferons. They increase the efficiency of macrophages. Lymphokines, complement and interferons, they increase the efficiency of macrophages, and the activated macrophages are not antigen specific. The activated macrophages, they secrete many biologically active substances. Those substances are hydrolytic enzymes, binding proteins like fibronectin and transferrin, tumor necrosis factor, colony stimulating factors and interleukin 1. Interleukin 1 here, it is a endogenous pyrogen, that means it raises the body temperature, and IL-1 in turn it induces synthesis of IL-2 by the T cells. These are the features of activated macrophages. Next one is dendritic cells. The dendritic cells are discovered by Ralph Steinman in 1970. These are the major professional antigen presenting cells. Dendritic cells are called as major professional antigen presenting cells. They are highly pleomorphic, that means exist in many shapes with a small central body and long needle-like projections like extensions resemble to that of dendrites of nerve cells. That is why they are called as dendritic cells. They are present in the peripheral blood and peripheral lymphoid organs. These dendritic cells, they arise from both myeloid and lymphoid lineages of hematopoietic stem cells. These dendritic cells, they possess the MSC class 2. Dendritic cells express the receptor, that is a MSC class 2, and along with the co-stimulatory signals like B7 and CD28. MSC class 2 is expressed along with this co-stimulatory signals. These co-stimulatory signals are required for the binding and activation of Th cells, because the dendritic cells majorly they function as a antigen presenting cells. They present the antigen to the T cell. MSC class 2 expression occurs on their surface along with these co-stimulatory signals like B7 and CD28. Functions of dendritic cell is these dendritic cells they capture the antigen at one location and present this processed antigen in the other location. The immature dendritic cells they engulf the antigen by means of three ways. Immature dendritic cells engulf the antigen by means of three ways, that is phagocytosis, another one is receptor mediated endocytosis, R for receptor, M for mediated, this E for endocytosis, and another one is pinocytosis. Pinocytosis is the taking of the liquids. Phagocytosis is similar to that of capturing the organisms or the solid particles, and receptor mediated endocytosis occurs only when they are bound to specific receptors, that is called receptor mediated endocytosis. So, the immature dendritic cells they engulf the antigen by the means of these three ways. By the process of maturation, the mature dendritic cells now they shift from antigen capturing to the presentation of antigen to the T cells. The immature dendritic cells are now shifted to the antigen presentation to the T cells that after maturation process. That means dendritic cells now express the co-stimulatory signals which are required for TH cell activation, that is required for naive T cells to mount an immune response. The mature dendritic cells now they express co-stimulatory signals for the activation of naive T cells. After activation, now the dendritic cells they leave the peripheral tissues, they enter into the blood and they migrate into the secondary lymphoid organs where the T cells are present. The major function of these is a antigen capturing and a antigen presentation. So, this is all about the cells of myeloid lineage. In the cells of myeloid lineage, the important cells that have a role in the immune system are the granulocytes. The granulocytes are basophils, neutrophils and eosinophils. Another one is a granulocyte, that is a monocytes, which later differentiated into macrophages and dendritic cells. Mast cell which shares the features with the basophil. In the next video, I will explain the cells that arise from the common lymphoid progenitor, that are lymphocytes. Thank you for watching this video.